Okay. First, I want to thank the organizers for inviting me, and I want to apologize for not being there as I intended to be there, but I had problems with biology, so which you might consider an extreme value problem in biology. So I couldn't come, but I'm glad to be able to present the talk at least on Zoom. So I thank the organizers for that. So the topic of the talk is a problem in, exactly as the title of the conference is, a problem in extreme value statistics, namely the thymic selection of T cells. T cells are cells that are part of the immune system, and they, as we'll explain in a minute, are selected in the thymus so that they have the right properties in order to be able to defend the body without, in fact, attacking the body. That's the question. This is work that was done a while ago with my longtime collaborator, Herbie Levine, who's there. And at the time was at Rice, is now at Northeastern, and his former student, Jason George, who is an MD, PhD, and has a PhD in physics and an MD, and he's currently at Texas A&M. And this is stuff that appeared in PNAS a while ago. All right, so what's the problem? So you have these T cells, and they're responsible for detecting foreign peptides, foreign proteins, and initiating an immune response. And they have to be able to do this without attacking the self-peptides, because that's what gives rise to autoimmune diseases, when the body attacks itself. So they have to be taught somehow. They somehow have to figure out, when they see this protein, is this something that's part of the body, or is this something that came from outside and needs to be dealt with? And so there's been a lot of work in trying to lay out the steps and how this all works. And part of the process is as the biologists have told us, is a process called negative selection. So in the thymus, you generate a wide random set of T cells, and then they're exposed to not all the self-peptides, because there's just too many of them, but a sample of them, a fairly rich sample of them. And now the idea is that any T cell receptor, which is the part of the T cell that actually binds to things, if it binds too strongly to a self-peptide, because the things that's exposed to are self-peptides, and if it binds to any self-peptide too strongly, then that's a dangerous T cell, and we want to get rid of it. And so, in the end of the day, those T cells that survive selection, in other words, they don't attack the body too strongly, these are the ones that the body multiplies and then sends out to do the work. And then the question, so we have a problem is, okay, they're supposed to not recognize self-peptides, and they're supposed to, however, react to other peptides. So what's being learned here? So you might imagine that there's some kind of pattern recognition going on, that there's some, magic code somewhere hidden. And if these T cells can just figure out how to decode the code, then that would be a good way to figure out how to identify friend versus foe. But that assumes that there's actually a pattern here, that there's some way of distinguishing a priori the set of self-peptides from the set of other peptides that the body encounters. And one of the ways to see how hard this problem is that if you look at the peptides associated with a cancer cell, the peptides are bound to this major histocompatibility complex, this MHC molecule, but that doesn't matter. But the peptide associated with the cancer cell, and many times only differs by one amino acid from a native peptide, because the cancer arose from some kind of random mutation. So it maybe differs by one, maybe two. And yet we want, and often, and we know that not rarely, the body is able to recognize this, what's called the neoantigen, as being a foreign protein, even though it's 90% similar, identical to a self-peptide. So that tells you how complicated this problem is. And so when we started going about this, what we assumed, and in fact, is that there is no pattern to learn. Rather, the negative selection, what it actually does, it just learns about the self-peptides it's trained on. Whatever self-peptides it sees, it recognizes them and just assumes that everything else is foreign. And in fact, this is consistent with a later work by Greenbaum and Bialik and others who actually tried to construct a statistical ensemble of self-peptides, and by that reproduced the right first and second order first and second moments, correlations between the peptides in the various positions. And they found out that, in fact, foreign and self-peptides have nearly identical statistics in the context of the kinds of models they were building, of maximum entropy type models. So the way we started working on this is there was a model by Chakrabarty and collaborators called the MJ model. And the model assumes that is a model for the binding energy between a TCR, this T cell receptor, which we're going to call T, and so the particular one is T sub J, say, and this peptide MHC complex, which we're going to call M. And it's given by, in this Chakrabarty model, the energy is a sum over the different I's, the different amino acids along the TCR. There are 10 spots in the TCR, so there are 10 different amino acids belonging to the TCR, and there are 10 different amino acids belonging to the peptide, and there's an interaction matrix which is assumed, which is a function of which amino acid sits on the TCR and which amino acid sits on the peptide. So and then it's just a sum of those interactions, so there's no crosstalk between different sites on the TCR. So each of these NT TCRs is characterized by 10, K is 10 in our model, that's fairly correct, amino acids, so which are T super J sub I running from 1 to K, where K is 10, and they're taken from the set of 20 different naturally occurring amino acids. And then each of the N peptides, ML, which are in the training set, they're the ones that the TCR is being exposed to, that are also characterized, each one, by a set of 10 amino acids, MLI. Okay, and as I said, this MJ matrix is some specific numerically fit to some set of data, matrix of interaction strengths between the TCR amino acid and a specific peptide amino acid. In fact, it's a symmetric matrix. And it's independent, its interaction is independent of the position along the TCR. It's independent of I, it's just a sum over the piecewise interactions of the 10 different sites. Okay. So this is the MJ model. In fact, there's two kinds of selection that go on. There's a positive selection and a negative selection. And as opposed to, this is a slight detail, divergence from the MJ model, the original MJ model, we're going to assume, because that seems to be what's biologically true, is that the training sets are different, because in fact, these two kinds of selections happen in sequence rather than together. What are the two kinds of selection? I talked about the negative selection, we'll come back to that in a second, but there's also a positive selection, because if you had a TCR which just didn't interact with anybody, then it would survive selection, but it is not, you don't want to have to devote your energy to producing these kinds of TCRs because they're not going to recognize anybody. So that doesn't help you. You don't get any benefit from TCRs that are just so totally weakly interacting that they just don't talk to anybody. So a TCR is rejected if, for the positive training set, the maximum energy of all of the peptides, the binding between this particular TCR, TJ, and all of the different peptides, if that binding energy is too weak, well, then we're not interested and we're going to throw that out. And then the negative selection is, again, if the maximum interaction energy with any given of the negative peptides, the Qs, is greater than some threshold. So if the TCR reacts with anything too strongly, any self-peptide too strongly, then we also throw that out. And so the final result is in order to make it into the body, the TCR has to survive both this positive selection, not be overall too weak, but not react too strongly against any one self-peptide. All right. And now we want to do some math. And so we consider a given TCR-T. Now, statistically speaking, the binding energy of T with a given peptide, a given random peptide, is a sum of K terms, which are essentially random because each of the 10 amino acids is chosen from one of 20 amino acids, and it's the sum of these numbers. So basically, it's a random Gaussian, you can think of it as a random Gaussian variable with some fixed variance, mu t, and mean mu t, and variance sigma t squared, which depends on the actual amino acids comprising the T. So, because it happens that some amino acids are weak in this MJ model, or more weakly interacting than others. So it depends on the actual mean and variance for the given amino acids in the T. And okay. So now what we need to do, as we saw here, is we need to calculate the probability of the max, the distribution of the max of the energies with the positive and the energies with the negatives, and we want it to be between these two limits, EP and EN. So this is just the maximum value of a Gaussian distribution over either the P or the Q training sets are given by the extreme value statistics of a Gaussian variable. And this was worked out back in the good old days, in the prehistory, in the actual birth of the field. The first papers on extreme value statistics was written by Tippett and Fisher, and the major problem they were looking at, in fact, even though they did extreme value statistics in all its generality and the different classes of the story, in fact, the thing they mostly studied happened to be in the extreme value statistics of the Gaussian variable, and it's in the Gumbel universality class, and so each of the EP and the EQ are the maximum is given by a Gumbel distribution. Okay, so, and now the Gumbel distribution is this exponential, double exponential, e to the minus whatever, and the width of the distribution, the Gumbel distribution is characterized, first of all, by a mean, that this is this En, which depends on n. It's logarithmically dependent on n. And the width is also logarithmically dependent on n. And this number n naught happens to satisfy this particular equation. N naught squared is 4 pi log n over n naught. And that's what was derived by Tippett and Fisher back in the day. Okay, so now we know the full answer. The chance to survive positive selection is given by the cumulative distribution function at one end of the Gumbel distribution. So the probability to survive positive selection is 1 minus e to the minus e to the minus, with EP there, and the chance to survive negative selection is just the other end, so it's just the CDF, it's e to the minus e to the minus. Okay. Now, so in order to survive both selections, it's just the product. So now we have worked out what is the probability, what is the survival distribution of the of surviving both positive and negative selection. And it's just this exercise in extreme value statistics. Okay, so now what we did to test that this all works, we take we just at random picked a single t, which happened to be given by that. And we took the difference between the positive and negative thresholds to be 5 in the right units that the MJ matrix is expressed in, calculated the overall survival probability for different values of N, because obviously everything depends on how many different peptides there are. And I remind you is the number of peptides both in the positive selection group and in the negative selection group. So, and here the dashed lines are the exact results and the solid lines are the theory, and it works fairly well. The reason it doesn't work perfectly is only because the convergence to the Gumbel distribution for the case of Gaussians is extremely, extremely slowly. It's logarithmic. And in fact, I was so troubled by this, I wrote a paper about how to accelerate the convergence, looking at the convergence properties and seeing if one can find better approximations. But that's neither here nor there. The theory works. So this is all very straightforward. It had to work, and it does work. All right. And now so far we're talking about a given single TCR. And as I said, this probability depends on these two numbers. It depends on the mean interaction energy, mu t, and its variance, sigma t, which is basically the mean and the variance of a given row or column of the MJ matrix. And now we want to average over all TCRs. So, well, okay, so if you think about mu t is an average over a column of this matrix, and so again, it's basically Gaussian by the central limit theorem, and you can calculate the mean and you can calculate the variance. These are just properties of the numerical MJ matrix that Chakrabarty had invented. Okay, now there's a slight difficulty because sigma t is also a Gaussianly distributed thing, that's true, but it's strongly coupled with mu t. As you see, because here we just plotted a point spread of sigma t versus mu t, and basically to zeroth approximation, the two are linearly related. So that's what we're going to assume. We're just going to take that sigma t is given by this linear relation with mu t, and assuming that, we can now average over the distribution of mu t, which is this Gaussian distribution. And when we do all that, you get the following results. Again, the quality is not perfect, but it's quite acceptable. And so we have calculated the probability of surviving as a function, say, of EP for a fixed difference between EP and EN. Okay, so that was a nice exercise. All right. Now we can ask a second question. And it turns out that this second question is the crucial question, which is, what is the contribution of an individual self-peptide to the negative selection process, right? I mean, we know that we're going to throw out a T if it interacts too strongly with any given peptide. And we're going to throw out another different T if it interacts too strongly with any given peptide. So the question is, well, if all of them are interacting very strongly with there's one self-peptide out there that with huge interactions, then everybody's getting thrown out by that guy. So the question is, how do the different, you would hope that the different self-peptides each contribute more or less equally to this selection process. So now, just as mu t governs the fraction of peptides that T recognizes, similarly the mu q, the mean, governs the fraction of TCRs that recognize Q. It's just the same thing turned upside down. And so similar to what we did before, with a fraction of TCRs that recognize Q is given by a Gaussian distribution, where the phi is the probability integral. It's the CDF of the Gaussian. Okay. And now the most potent one is the one with the, because this is an increasing function of x, therefore it's a decreasing function of mu. Oh, sorry. And now, so the most potent peptide is the one with the largest mu. We just look over the data set and we figure out which is the one that's the maximum, and it has a value of 42.77 for our selection of 10 to the 4th peptides that we generated, and its sigma q is 4.09. plugging that into the formula, and if you choose the threshold such that you get 50% selection so that half the TCRs are allowed to survive, in fact, what you find out that RQ is 0.428, which is actually very close to the true value of the predicted value was 0.428, and the true value for this particular peptide was 0.431. In other words, this one peptide is responsible for the elimination of 86% of all the TCRs in the entire population. Right? You look at all the TCR, you've adjusted the parameters so that you're throwing out half the TCRs, and this peptide is responsible for 86% of that because they eliminated 43% of the 50%, which is, so you have 86% are killed by this one peptide. So that's truly ridiculous somehow, because then everything depends on whether this one peptide is in your training set or not. And that would be horrible. Clearly, that's not where you want to be. So now you actually can calculate what this mu Q max is if you felt like it, but in fact, the problem, and we did it, and shows that the problem is systemic. It's not the question of just bad choice of parameters. It happens generically, which means that it's time to cut and run. This model is clearly wrong. This is clearly not giving us reasonable results because it's saying that all the selection is due to basically one single peptide in the training set, which is sort of means that everybody else is just, sitting there doing nothing. So we have to invent a new model. Now, so the next model we invented, and there's only 3, so you don't have to worry about it. It's a finite number. We called the position-independent random affinity model. So it's a modification, as you'll see, of the basic ideas of the MJ model. Again, we have that the binding energy is a sum over K of numbers, but now the numbers are not dependent on this MJ matrix. In fact, they're random numbers, just random Gaussian numbers. And the random number depends on just the peptide T, the TCR T, and the amino acid that it's interacting with. So each TCR interacts differently with each of the amino acids. So a TCR is now is characterized by this set of 20 different values of psi A, psi Q. The interaction of this particular TCR with each one of the different amino acids it might see in a peptide. And notice again that it's independent of position. It's just a linear sum over position. And now we want to analyze this model. And this model is a little trickier. And it turns out that what's the critical thing here is the role of multiplicity of the amino acids in a given peptide. Because each of them have the same mean, but the variance varies tremendously wildly. Because if you have an all-alanine peptide, a peptide which is this, A, 10 different A's, then the variance of the binding energy for that would be 100, as opposed to 10 for a peptide with no repeats. Right? When you have a peptide with no repeats, the variance is the sum of the variances, and each of the variances is 1 because these are standard Gaussian random numbers, because we don't care about the units anymore. We've gone into physics mode. But if you had identical proteins, if you looked at the difference between an all-alanine peptide with an all-something else peptide, the variances of the ones that are all the same would be 10 times larger. So now you have to do the analysis for each different class, a multiplicity class, and the expected number of TCRs that recognize a peptide of the multiplicity class is the same formula, except that now sigma depends on the class. And it turns out when you have a finite population, like 10 to the 4th, you can figure out what is the most potent peptide is the one with the largest sigma C, and that one, i.e. the most amount of duplications. And for a population of 10 to the 4th peptides. It's a peptide with 3 singletons, 1 doubleton, and 1 thing with multiplicity 5. You can work out that and figure out what is the potency of that. And it turns out this one single peptide, which has this particular kind of multiplicities, is responsible for 2% of the selection of the TCRs, which is way, way better than 43%, but it still means that, in fact, roughly speaking, only 100 of the peptides are contributing out of 10 to the 4th, which is still not really where you want to be. So let's go back to the drawing board again, and we come up with our final model, which we call the random interaction between cell receptor and epitope model. And if you notice that the acrostic is RICE, and the fact that 2 of these people worked at Rice University, that had nothing to do with it. This was completely generated independently of that. But okay, so here, what's the difference? Here, the chi, before the chi only depended on the TCR. There was a set of chis for each TCR and each kind of amino acid. Here it also depends on position. So the chi is a function of two scripts. One is the position and one is the type of amino acid. So if this TCR interacts differently with alanine in position 1 than it does with alanine in position 3. Okay. And again, you can work out what the probability of selection of a single self-peptide by a single TCR is. It's given by that. And the probability of a TCR to survive selection is essentially this to the nth power, so it's just e to the minus nRq, because RQ is small, because otherwise it would be 0. And the number, since n is big, and the number of TCRs recognized by a given peptide S is so therefore Poisson distributed with mean nRq. All right. Now, if the number of TCRs is much greater than n, the number of self-peptides, and nRq is a number of order unity, but nTRq is much, much bigger than 1, and the distribution of S is approximately Gaussian with mean invariance given by nTRq, which is a large number. And so therefore, again, we're back to Gaussian extreme value statistics. Looking for the most potent peptide is just the largest value of this number. And there, so you get, again, this logarithmic dependence on nn. And so what you find out is for NT equals 5 and NP equals 10 to the 4th, 50% selection, what you find out is the most potent peptide in this third model, in fact, is it only rules out 17, which is a tiny fraction. So we finally got a model which gives us something that at least is semi-reasonable. And so in summary, we can summarize these results by these 3 curves. For the original sequential MJ model, what we found is that the cumulative, that assuming the asymptotic selection is 50%, in fact, once you've done the most important self-peptide, you've eliminated the 50%. You don't have to look at anybody else. It's not quite so bad with this intermediate model, this pure model, which was position independent. But what if you go to a position dependent model, in fact, then all the different peptides are contributing to the process. And then you can go and ask, and the question, the burning question is what happens to when you have a self-mutated self-peptide, a point mutated self-peptide. So you've gotten to where the probability of recognizing of a given TCR recognizing a peptide is 50%. What do you do about a point mutated self-peptide? Is it recognizing it or not? And the point is that the model is learning, and you find out, you can do the calculation and find out that in fact the difference between the probability of recognizing a random peptide, something that has nothing to do with the training set, and something that is in fact identical to the training set except one amino acid substitution, in fact, the difference between that is never greater than a factor of 3.5. So in fact, while a point mutated self-peptide is not a random peptide, it's only 3 times as likely to survive being recognized by the set of TCRs, which is a reasonably small price to pay. So the model here actually is learning only evidently the specific self-peptides it encounters. And so even when you single mutate a self-peptide, there's some reasonable chance of being recognized as foreign and therefore the body attacking it as a tumor cell. So I thank you for your attention, and that's it.